Researchers have successfully determined the mechanisms behind how dietary restriction can effectively slow down the aging process of the brain and extend an individual's lifespan. Greetings and salutations to everyone. Today's video focuses on the connection between dietary restriction, aging delay, and the deceleration of neurodegenerative diseases. Researchers from the Buck Institute for Research on Aging have conducted a study that highlights the significance of a gene known as mustard, MTD, in fruit flies and oxidation resistance 1, OXR1, in humans and mice. This gene plays a crucial role in safeguarding cells against oxidative damage, particularly in the brain. It directly impacts the retromer complex, responsible for recycling cellular proteins and lipids, which is essential for maintaining cellular health. Dysfunction of the retromer complex has been linked to age-related neurodegenerative diseases. The research team discovered that MTD/OXR1 is instrumental in preserving retromer function, thereby supporting neuronal function, promoting healthy brain aging, and contributing to the extension of lifespan observed in dietary restriction. Without further delay, let us dive into our discussion. According to the study's first author, Kenneth Wilson, PhD, a postdoc at Buck Institute, individuals who limit their food intake often consider its impact on their digestive system or body fat, but rarely consider its effects on the brain. However, the study, published in Nature Communications on January 11, 2024, reveals that this particular gene plays a crucial role in brain function. By studying both fruit flies and human cells, the team not only revealed a comprehensive cellular process that explains how dietary restriction can effectively postpone the aging process and decelerate the advancement of neurodegenerative disorders, but they also pinpointed potential targets for therapeutic intervention to combat aging and age-related neurodegenerative diseases. According to co-senior author of the study, Professor Pankaj Kapahi, PhD, a specific response within neurons has been discovered that is responsible for the neuroprotective benefits of dietary restriction. The study suggests that strategies like intermittent fasting or caloric restriction, which restrict nutrient intake, can potentially increase the expression of this gene and thereby enhance its protective effects. The gene serves as a crucial factor in safeguarding the brain against the effects of aging and neurological disorders, explained Dr. Lisa Ellaby, co-senior author of the study and a professor at Buck Institute. The research team has previously uncovered mechanisms that enhance both lifespan and health span through dietary restriction. However, the diverse range of responses to calorie reduction among individuals and tissues indicates the existence of numerous undiscovered processes at work. The objective of this project was to unravel the reasons behind the varying responses to diets among different individuals. To initiate their study, the team conducted scans on approximately 200 strains of flies, each possessing distinct genetic compositions. These flies were subjected to two distinct feeding regimens, a standard diet and a restricted diet comprising only 10% of the usual nutritional intake. Through their investigation, the researchers pinpointed 5 genes that harbored specific variations that exerted a substantial impact on lifespan when subjected to dietary restriction. Remarkably, 2 of these genes exhibited analogous counterparts within the realm of human genetics. The research team made the decision to thoroughly investigate a specific gene known as mustard, MT, in fruit flies and oxidation resistance 1-inch, OXR1, in humans and mice. This gene plays a crucial role in safeguarding cells against oxidative damage, but its exact mechanism of action remained unclear. In humans, the absence of OXR1 leads to severe neurological impairments and premature death. Conversely, in mice, an excess of OXR1 has been shown to enhance survival in a model of amyotrophic lateral sclerosis, ALS. In order to comprehend how a gene that primarily functions in neurons can impact overall lifespan, the team conducted a comprehensive series of tests. Their investigations revealed that OXR1 directly influences a complex known as the retromer, which consists of a group of proteins essential for recycling cellular proteins and lipids. The retromer plays a critical role in neurons as it determines the destiny of all proteins that enter the cell, explained Wilson. Dysfunction of the retromer has been linked to age-related neurodegenerative disorders, such as Alzheimer's and Parkinson's diseases, which interestingly are mitigated by dietary restrictions. The findings of their study provided a comprehensive narrative on how brain aging is decelerated through dietary restriction, thanks to the involvement of MTD/OXR1 in the preservation of the retromer. Kapahi expressed, "The significance of this research lies in its demonstration of the crucial role played by the retromer pathway, responsible for recycling cellular proteins in safeguarding neurons during periods of nutrient scarcity." The research team discovered that MTD/OXR1 not only maintains the functionality of the retromer, but also plays an essential role in promoting neuronal health, supporting healthy brain aging, and extending lifespan in individuals practicing dietary restriction. According to Wilson, the gene is being influenced by diet. When you consume fewer calories, you are actually promoting the efficient sorting of proteins within your cells. This is due to the fact that your cells are increasing the expression of OXR1. Researchers discovered that increasing MTD levels in flies resulted in an extended lifespan, which has led them to hypothesize that elevated OXR1 expression in humans could potentially lengthen life. Ellaby stated that their next objective is to pinpoint particular substances that can raise OXR1 levels as individuals age, thereby delaying the aging process in the brain. Wilson expressed his hope that through this, we can gain further insights into the underlying causes of brain degeneration. The impact of diet extends to every function within your body, he stated. This research reinforces the importance of adopting a nutritious diet, as the food you consume has far-reaching effects that may surpass your awareness. The study involved various Buck researchers, including Sudipta Bar, Enrique Carrera, Brian Hodge, Tyler Hilsebeck, Joanna Bonds, George Brownridge III, Jennifer Beck, Jacob Rose, Melia Granath Panello, Christopher Nelson, Grace Kui, Akos Gerenser, Jian Feng Lan, Rachel Brem, and Birgit Schilling. Financial support for this research was provided by the National Institutes of Health, NIH, the Larry L. Hillbloom Foundation, and the National Centers of Competence in Research, NCCR. Pankaj Kapahi serves as the founder and a member of the Scientific Advisory Board at Juvify Bio, while the other authors have no conflicts of interest. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.